Thank you for participating in this study challenge. Here's a question from the USABO Semifinal exam June 24, 2020. Please drop a like, comment, share this out, and subscribe as we continue to do USABO together. Click the link below if you are looking for biology help. To quantify cell division and proliferation, the experiment utilizes the dilution of CFSE fluorescence. The main idea is that the label gets diluted as the cells divide. Looking at panel C, there are high counts at lower fluorescent intensities relative to the control. This is what you would expect when the dendritic cells communicate with the T cells by presenting antigen. If the cells are deficient in dendritic cells, then you would expect infected and uninfected mice to display the same panel. Thank you for participating in this study challenge.